This will be part 3 of our lesson series on life evolving. And we're going to try to figure out how it is that little blue bacteria had such a survival advantage compared to their red competitors. And the story we're going to tell is one of gene duplication. Now, gene duplications create new evolutionary possibilities. Let's recap the process of DNA duplication. So bacteria have a circular chromosome, and there might be, a few thousand genes encoded in this circular piece of DNA. Remember, a gene is that sequence of DNA that is a recipe to build one particular protein. Of course, when the bacteria is preparing for cell division, it's got to duplicate its chromosome. And so here we see the complementary letters finding their matching partners on the 2 DNA strands. When that process is complete, then we have 2 identical circular chromosomes, although there may be mutations here, new mutations, but one individual will get this chromosome, and then the other daughter cell will get this chromosome, and the parent will have divided into 2 individuals. Now, a gene duplication is a really interesting kind of mutation. So here, let's take a look at gene X. So any particular gene here is one of the many genes in this chromosome. Well, when the whole chromosome was duplicated, that gene was copied twice. So there really should just be one gene X here in the duplicated chromosome, but the gene itself was duplicated as the DNA was being copied. So now the offspring is going to have 2 of the same versions of the gene, 2 genes that encode the same protein. Now this is interesting because one of these genes here, the offspring can preserve that recipe to make the original functional protein. But now it has a second copy of that gene. And this means that this duplicate gene is free to accumulate mutations without loss of the original protein. So you see, it's gene duplications sort of provide living things with a chance to experiment with new genetic information. So gene X is the recipe to make some useful protein doing something useful. But now what if you played around with that recipe? Could you make some other new protein that can do something else new and useful? That's what gene duplications do for life on Earth. Okay, let's return to our story then. Here we have the red bacteria eating the green food. Now, why can they eat the green food? Let's take a look down here. They can eat the green food because they have a protein that is specialized to import the green food molecule into the cell. Like all proteins, this transport protein has a corresponding recipe, a gene. We'll call that the transporter gene. Now, when the cell copied its DNA in preparation for reproduction, the entire transporter gene was accidentally duplicated. And this is an important kind of mutation. So because both of these genes are still functional, the cell might have actually just made extra of the transporter proteins and inserted them into the cell membrane. But let's say that having this extra functional duplicate gene does not produce a survival advantage. So this individual might import a few more green food molecules, but it doesn't give it really an advantage compared to a cell that only has one transporter gene. However, look at what happens in the next generation. So during the next round of DNA duplication, a mutation occurs in the duplicate transporter gene. That's the little arrow right there. Now, this first mutation is random in the sense that the bacteria cannot steer mutations to be useful. So it's not like this parent was sort of, while duplicating DNA, and kind of saying to itself, hey, let's throw some mutations in this particular gene at these locations, and let's try to make some new useful protein. No. These are random errors during DNA duplication. But in this case, the sequence was changed in this particular gene, and we're going to see it had an effect on the corresponding protein. So the altered DNA sequence changed the amino acid sequence of the transporter protein, which changed the shape of the protein. And the new transporter protein is not able to import the green food molecule. The protein is non-functional. But the individual is still able to import the green food because the original gene is intact. So see, already we're seeing the significance of this. By having 2 of these transporter genes, one of them can make the functional protein and the other can accumulate mutations even as in this case, it creates a non-functional protein. Notice this individual does not have a survival advantage. They have two transporter genes, one of which a mutation has knocked out the function of that transporter, but it has no advantage as a result of having this extra gene and having that mutation. It eats green food just like the original red competitors. However, now we get to the final stage of the story. Another round of DNA duplication produces a second mutation in the duplicate transporter gene. This time, the altered sequence changes the shape of the protein in a functional way. The new transporter protein can do something new, import the orange food molecule, a new source of food. Notice then, now we have an individual capable of eating a new food source, and that is a survival advantage that leads to a reproductive advantage. So now you get this mutant, the one with a duplicate transporter gene with a couple of random mutations, but now can eat the orange food. That's the trait that gives them the survival advantage, and now you get the evolution of that trait in the population. So the trait becomes more common in future generations. We would say the current environment of green food scarcity selects the individuals capable of eating the orange food. The selected individuals survive and reproduce more successfully. And remember, we said we can think of selection in the opposite way, that the environment is punishing individuals that don't have this new trait. So kind of like a filter, the new orange food eating trait is the advantageous trait, and if you don't have that trait, you will starve. If the green food molecule were always plentiful in the environment, the ability to eat orange food molecules is not an advantage, assuming the foods are equivalent. So if there was no scarcity of green food, then being able to eat the orange food is not an advantage. And if this is not an advantage, then the blue bacteria will not increase in frequency in the population. So the orange food eating trait might happen in the population, but it won't increase in the population if it doesn't provide an advantage in the current environment. No evolution without an advantage. So this is an important feature of evolution. Evolution cannot see into the future. Bacteria cannot evolve orange food-eating traits as insurance against the possibility that green food molecules will disappear in the future. So natural selection is just operating in the current generation, but it, of course, continues to operate in every succeeding generation. But evolution cannot see into the future and equip a species with some adaptation they don't need now, but maybe could need in the future. Evolution doesn't work that way. Evolution equips creatures and species with adaptations to deal with the current environment. So a couple of important points about this evolutionary change. The mutations that produce the variation are random. So living things can't steer mutations to be just beneficial for them. Many mutations mess things up. On the other hand, natural selection is non-random. And by non-random, we mean that individuals do not have equal chances of survival. Because there are differences that make a difference to survival, individuals have unequal chances of survival. So it's not random who gets to survive. It's not random at all. It's the individuals with the best fit traits get to survive and pass on those traits. In the next lesson, we're going to generalize this concept of how new proteins can evolve to make the claim that all functional proteins have this kind of evolutionary history. That the reason a protein has a function is because it evolved to perform that function.